My name is William Milson, and the title of this presentation is The Long-Term Effects of Crop Diversification on Soil Fungal Communities. Crop diversification practices are often applied to enhance soil health and plant productivity, but little is known about the biological mechanisms behind their effects. Crop diversification can modify the soil environment in different ways, including a change in carbon and nutrient inputs. We expect soil microbial communities to be sensitive to these changes, possibly revealing changes in their diversity and composition. Our goal was to evaluate the long-term response of soil fungal communities to two temporal crop diversification strategies, winter cover crops and crop rotation. In the workflow for these studies, we obtained and stabilized soil samples in the field. We extracted soil DNA using standardized test kits, and we analyzed fungal amplicons using high-throughput sequencing and bioinformatics tools. The first study consisted of a 4-crop horticulture rotation, where we analyzed the effect of 4 different cover crop compositions, together with a no cover crop control. Our results showed only minor effects of cover crops in soil fungal communities. On the left, we can see that fungal phylogenetic diversity was not affected by the cover crop treatments. On the right, a principal coordinates analysis shows no treatment-based grouping in terms of phylogenetic composition, even though the cover crop effect was significant. The second study evaluated 5 crop rotation systems with different plant species diversity and analyzed under conventional tillage and no tillage. To standardize conditions and focus on long-term crop rotation effects, sampling was carried out when all rotations were under corn cropping. Long-term effects of crop rotation were clearer than cover crop effects. The figure on the left shows that even though no significant treatment effects were detected, the response of fungal phylogenetic diversity showed opposite trends between no-till and conventional tillage. On the right, the principal coordinates analysis shows a clear effect of tillage practices, and within each of them, a similar gradient of crop rotation effect, with soils under continuous corn cropping presenting the most distinct fungal communities. In both cases, the number of species in the rotation seems to be less determinant than the types of crop species included. Our results showed that 7 years of cover crops in horticultural crops did not have a strong influence on soil fungal communities. On the other hand, over 30 years of crop rotation practices shaped soil fungal communities, but not more strongly than tillage practices. Our next steps included deeper analysis of both fungal and bacterial communities, their interactions, and their repercussion in soil health and provision of ecosystem services.